my name is Mullikin, and today I'm going to tell you about my favorite cell organelle, the centriole. Organelles are parts of a cell that have specific functions, such as protein production or energy production. Before we look at what a centriole does, let's understand its structure. The centriole is a wonderfully symmetric, cylindrical arrangement of 9 microtubule triplets, each connected to a central hub by a spoke and to each other by linkers. Head on, it kind of looks like this rotelli pasta. The outermost microtubule of each triplet is slightly shorter than the other two, creating a distinction between the proximal and distal ends. The proximal end is embedded in a massive protein called the paracentriolar material. Each cell contains a pair of centrioles. While the cell is growing and duplicating its DNA in preparation for cell division, the centriole self-replicates, creating a daughter centriole. The mother and daughter centriole remain aligned at a 90-degree angle, and surprisingly, if centrioles are destroyed, as long as the necessary proteins are available, centrioles can actually form without the presence of a mother centriole. Now for function. The two most important roles of centrioles are anchoring cilia and flagella and organizing microtubules. Cilia are short extensions and flagella are longer extensions that help to mobilize the cell. When a centriole acts as an anchor to a cilium or a flagellum, it is called a basal body, and only mother centrioles can be basal bodies. Cells also gather specific proteins and organize them into microtubules, the largest and most rigid of cell fibers. This provides a cell with a frame that gives it shape and creates little railways for transport inside the cell. Centrioles also help to build the microtubules that make up the mitotic spindle. These fibers connect to chromosomes, or bundles of DNA, and wrench them apart during cell division so that the two cells produced end up with essentially identical copies of genetic information. But higher plants and fungus cells do not actually contain centrioles, so how do they produce new cells and grow? They actually can use a different, more spontaneous method to build microtubules. And even more fascinating is that in animal cells, when centrioles are destroyed, the cells can still divide. For example, in Drosophila, fruit flies, when centrioles were destroyed by laser ablation, the organisms could still grow, but they couldn't survive because there were no basal bodies for cells that needed cilia and flagella. So centrioles are incredibly intriguing because they've been conserved since almost the beginning of evolution, as can be seen in that all animal cells that have them have centrioles with essentially the same structure, but yet they're not expressly necessary for cell division. So this raises the questions: what is the exact benefit of centrioles in cell division? How do they make the process more efficient on a molecular level? And how do cells account for their presence or absence at cell cycle checkpoints? With these questions in mind, I hope this lecture has piqued your curiosity about centrioles.